principle on and performance of electrophoresis as it applies to hemoglobin, to discuss the significance of performing hemoglobin electrophoresis. So basically electrophoresis is a method through which charged molecules in a solution, chiefly proteins and nucleic acids, migrate in response to an electrical field. The rate of migration through the electrical field, it depends on strength of the field, on the net charge, size and shape of the molecules, and also on the ionic strength, viscosity, temperature of the medium in which the molecules are moving. So as an analytical tool, electrophoresis is simple, rapid, and highly sensitive. It can also be used analytically to study the properties of single charged particles or mixtures of molecules. It can also be used preparatively as a separating technique. We will discuss the separating technique in the slides later on. So at any given pH exists in a solution as electrically charged species, either as a cation or anion. Ionizable groups present in the protein molecules are electrically charged, and as I told you earlier, they are either cations or anions. And at any given pH under the influence of an electrical field, these charged particles tend to migrate either toward a cathode or anode, depending on the nature of their net charge. So in many electrophoresis units, the gel is mounted between two buffer chambers containing separate electrodes, so that the only electrical connection between the two chambers is through the gel. So as in the picture you can see, these are the types of gels: tube gel, vertical slab gel, and the horizontal slab gel. So this electrokinetic process separates protein molecules or DNA through different procedures depending on the type and size of the molecule. The procedure differs in some way, but all need a source for the electrical charge, a support medium, and a buffer solution. So here you can see that gels are formed, as I told you earlier, in the tubes and slabs. The tubes are not used nowadays. As it is tricky to get a set of gels with identical length, hard to remove the gels from the tubes once they had been run. However, tube gels give a better resolution of the results, so we often choose them for protein electrophoresis. Sometimes, but not always. So in this picture you can see a model of an electrophoresis machine. So this is a horizontal or submarine gel, and different types of gels are used as a support medium for electrophoresis depending on which is more beneficial for us. Gel slabs, either horizontal, vertical, or vertical, enable many samples to be run simultaneously, and so are frequently used in laboratories. This is basically a horizontal gel type, which is also known as submarine gel, and it is showing electrodes here. These are the electrodes, the positive and the negative, at the two corners, and these are submerged in a buffer solution here, and in the gel in between. So these two electrodes are submerged between the buffer solution and the gel. So, significance of electrophoresis. In electrophoresis, chemicals used such as blood, proteins, DNA, or inorganic ions can be separated according to the differences in their mass or charge. Protein electrophoresis tests give a health practitioner a rough estimate of how much of each protein fraction is present and whether any abnormal proteins are present. So, we are going to discuss. So now you have an overall idea of what electrophoresis is all about. Now we are coming to towards our concerned instrument, which is the hemoglobin electrophoresis machine. And in this picture, you can see a gel slab, the electrolyte solution in which the electrolytes, the cathode and anode are submerged, and this is the hemoglobin electrophoresis machine. So hemoglobin, which is an oxygen carrying substance in the blood, is tested by this instrument. We separate and measure the different hemoglobin molecules depending upon their charge and quantity through electrophoresis. Electrophoresis is the means of separating hemoglobins, and it depends on the migration of hemoglobin molecules, again according to their charge and amount dissolved in the buffer. So this is all we are going to discuss in our later slides. So hemoglobin electrophoresis is of two types basically: the cellulose acetate at alkaline pH and the citrate agar electrophoresis at acidic pH. So the cellulose acetate at alkaline pH is simple, reliable and rapid. It is satisfactory for the detection of most common clinically important hemoglobin variants. Citrate agar electrophoresis basically separates hemoglobin fractions that migrate together on a cellulose acetate agar. All hemoglobin specimens that show an abnormal electrophoretic pattern in an alkaline media, that was the cellulose acetate, they should undergo the citrate agar electrophoresis. So the citrate agar electrophoresis at acidic pH permits the separation of major hemoglobins based on the relative mobilities of variant hemoglobins. So the pH of this citrate agar electrophoresis is around 6 to 6.2. So these are the two commonly used types of hemoglobin electrophoresis. One is the cellulose acetate, which operates at an alkaline pH, and the citrate agar, which runs at an acidic pH. And any abnormal protein which is detected by the cellulose acetate should undergo citrate agar electrophoresis for a more detailed, separation of the major hemoglobins. So here you can see the types of hemoglobins: AA, ASAA, which is the normal variant, AS and SS, and all of them showed specific movement patterns towards their respective anodes, so they can be easily recognized. So this is the migration of these hemoglobin variants. This is the AA, and this is the AS, and this is the SS. So the AA moved towards the anode, farthest towards the AA variant of the hemoglobin is most attracted by the anode, while the SS variant is least attracted by the anode. So now coming towards the principle of this test. So the principle in which this instrument works is same for both for both the types of which is the cellulose acetate and the citrate agar electrophoresis. Right now I'm explaining the electrophoresis with reference to the citrate agar plate. Usually cellulose acetate electrophoresis is done first to find out any abnormal hemoglobin. Then citrate agar electrophoresis is done as a follow-up for confirmation of detected abnormal hemoglobin. Excuse me. So the whole blood is centrifuged first. The hemosylate reagent hemolyzes the red blood cells and stabilizes the hemoglobin. We take only the packed cell volume for hemoglobin electrophoresis. Then very small quantity of this sample is applied to the citrate agar plate. All types of hemoglobin present in the sample are then separated by electrophoresis using citrate buffer and are stained. The patterns are scanned on a scanning densitometer, and the relative percentage of each band is determined. So electrophoresis is a procedure which enables the sorting out of molecules based on their size, as we discussed earlier. Using an electric field, molecules such as DNA can be made to move through a gel made of agar or polyacrylamide. The electric field consists of a negative charge at one end, which pushes the molecules through the gel, and a positive charge at the other end that pulls the molecules through the gel. The molecules being sorted are dispensed into a well in the gel material. The gel is placed in an electrophoresis chamber, which is then connected to a power source. When the electric current is applied, the larger molecules move more slowly through the gel, while the smaller molecules move faster. The different sized molecules form distinct bands on the gel. So these are some of the reagents we use while doing the hemoglobin electrophoresis. These are the electrophoresis chamber and the agarose gel casting tray, buffer, staining agent, which is a dye, a comb, a DNA ladder, and the sample to be separated. So these are the parts of a gel electrophoresis or the hemoglobin electrophoresis machine. This is the hardware. This is the power supply, and this is the gel tank. This is the casting tray, electrical leads, and the gel combs, and the cover. So the lower or acidic pH of the Titan 4 citrate agar allows it to be used as a primary screening procedure for detection of hemoglobin S, which is the sickle cell hemoglobin in the newborns, and as a confirmation for abnormal hemoglobin detected by alkaline electrophoresis. So up to 14 samples plus 2 controls can be run on each plate. Samples are easily applied with zip zone applicator. The citrate buffer is used as a chamber buffer for the citrate agar procedure. Buffer provides ions that carry a current and maintain pH at a relatively constant value. So basically, the citrate agar allows us to determine the quality and the quantity of the HbS in the newborns.